Hello and welcome to BioCode. In this video, we will see how and what are the uses of Fastq files within bioinformatics analysis. Why do we use them, and what are the features that are provided by Fastq files? Basically, Fastq files are the extension of the file system that is file format that is provided by FASTA format, which is basically the sequence to hold the particular sequence. For example, it could be a protein, gene, nucleotide, or whatever. So in that scenario, what really happens is that in FASTA files we only have the identifier of the sequence, and that could be the accession ID, the description, and then we have the sequence itself. But FastQ files has other FastQ files do have other information, such as information about the sequence reads and that sort of information, which we will explain in this video. But first of all, you need to understand what is basically sequence reads, and why do we actually need FastQ format at all. So basically, when we are trying to when we are trying to sequence a particular genome, that sequenced genome has to run over the sequencing technology. It could be utilized through PacBio, Illumina, or whatever different sort of sequencing technology that is available. So each of that technology generates thousands of billions of millions or billions of reads of a particular genome, and those reads have to be assembled into a shotgun, whole genome shotgun, and then constructed into a assembly itself, which obviously takes time as we go down the analysis. But first of all, in this video we only are covering the SRA, which means the sequence reads, and SRA is also meant to be a database, which means the sequence read archive. So basically, sequence reads are the actual reading of the sequence data, but that could be overlapping, which then we then assemble, and then we find the overlapping regions, and then we stick them together. But that is the topic of another video. But in this video, you just need to understand that sequence reads are individual base calls of the genome that we are trying to sequence, and that genome has to be converted into some sort of sequence data so we can understand it, because it is just a base call, it's just in binary, and we don't really understand it unless it gets converted into a format that is understood by the sequence analysis tools, or even by the humans themselves. So suppose that we have sequenced genome and we have received some sequence reads. So in this scenario, what really happens is that they are converted from base calling to FastQ files, or in other scenarios they are also known as FastQ files. So once they have been converted into FastQ files, they can be and they can be utilized in any sort of analysis that leads after the genomic sequence. But in this video, we are going to understand what is FastQ conversion, and what is basically the use of FastQ files. So FastQ files basically provide us information about each of the nucleotide, each of the base calls that are provided, and those base calls can be A, C, T, G, and N for the ununderstanding region, and or the non-understanding region of the particular genome that or particular region of that genome. So basically, it provides us the ability to pinpoint each of that nucleotide that is present between the sequence reads, and associate a particular score to that particular region, that particular nucleotide within the genome, so that we can utilize them in the next generation sequencing or other sort of analysis that goes within the genomic sequence, so that we can understand, okay, these regions of the genome actually are not as accurate as they should have been, and then the scientists rerun their genomic sequences so that they can find the accurate regions, because in most cases when we are doing genomic sequences, we find regions that are not accurate as we hypothesized, because there are so many errors within sequencing technologies, and it is not perfect yet. So up until we have based that FastQ files provide a sequence identifier, and the information is provided such as the technology that was used to run the sequencing, and the cluster that was formed for the sequencing reads, and the exact contents of the base calling that are converted from the BCL to FastQ format. For example, it could be from the binary to ACTGN. But it also provides a separator, which is a plus sign. Basically, we will see what the file looks like, but first of all you need to understand the theoretical background. So a plus sign is provided that actually separates the base quality scores from the actual sequence, and then we utilize Fred scores, Fred scores, which are basically 33 plus 33 encoded, base coded, and we utilize ASCII characters to represent those numerical quality scores of that particular region of the genome that we are actually visualizing in the FASTQ format. So there are three. We will actually see what the Ferret score looks like, and we do have a picture on that. So this is basically what it looks like if you will take a good look at it. Basically, there are 33 base ASCII characters, that each of the character that is utilized within FASTQ format has its own particular score, and this score is read by FastQ interpreting softwares that can understand, okay, these regions are lacking the accurate information, and these to have some good information based on the quality score that is associated by to each nucleotide region, and to each of these region has its own character, ASCII character, and that defines its quality. So it can be, as you see, it goes from 0 to 42 in total, but depending upon the technology that you are using, such as Illumina, Ion Torrent, PacBio, and Sanger, and each of these have their own probability error and ASCII score and their ASCII coding, and the actual characters are these are the characters which could be alphabetic, which could be symbolic, and which could be other capital or small or capital letters as well. So we have established that we also utilize the Ferret quality scores, which could be 33 plus 33 base scores, and there are multiple FASTQ variants, such as Sanger standard. Basically, there is not a particular standard that is utilized by all of these companies, all of these technologies. So they have their own variants of FASTQ format. Sanger has its own, Solexa and Illumina has its own, but they are not that different. But they are mostly similar, but just minor tweaks here and there, just to make those standards work with their own technology. So just to provide you a basic context of what is FASTQ format. FASTQ format is just a simple extension of FASTQ format, but only it provides the ability to store a numerical quality score according to each of the nucleotide present in the sequence or the sequence read. So it is just a plain single FASTQ format. Just it provides a quality score. So that is why we utilize the Q word. So it becomes FASTQ. That means FastQ or FastQ, that means quality. Q stands for quality here. And the Ferret score and the quality scores depends is basically assigned to each of the value that is present in the base, or the basically base means the nucleotide. So each of these values that are associated to each of these bases is done by Ferret software, and they are assigned, and then we do have the probability errors and that sort of information which we saw in the earlier picture. And these are the errors, basically P error, which are Ferret errors or probability errors. So now we can see what actually FastQ file actually looks like. It's the extension is FastQ or FastQ, and this is just a random files that we have created, and you can see. Let me just remove this information just to make it a little smaller, and okay, we will explain each of the context of this file, and we will also explain the usage of this particular file. So basically, and unlike fast format, which starts with a greater than sign, and then the ID goes over here, then there is a description over here, and then we have the sequence itself, right? So in this scenario, on FastQ scenario, we do not have such information like this. We do have sequence information like present over here. We do have the ID and the description that is present over here, and this is the score that is provided to each of the base call. But the orientation or the context of the file is a little different. Just there is a particular extension onto the FASTA format. So first of all, what you need to understand is that it starts unlike FASTA format, which used to start with the greater than sign. It starts with an at or the rate in some cases. So then we have the instrument information or the ID itself that was utilized in this base call or in this sequence read, then the run number, then the flow cell ID, the lane of the flow cell, the title of the flow cell, and the position x axis, y x position, the read information, if it is filtered or not, control number, and the sample number. That sort of information is provided in the FastQ. So now you can understand that FastQ file has a lot of information that can be utilized in a single file format as compared to FASTA format, which does not really have that much information. So what all of this means, what it does it look like, we do have an information on that too as well. And first of all, you need to understand that whenever we are actually utilizing thread quality scores, there are probability errors that goes with each of the base call, and these are the actual base call accuracy, and the quality scores that go with the 60 are the least, and they provide the best accuracy as compared to as the quality score goes down. So that means each of the base has its own score, and each of the base has its own accuracy. So if that means, so as long as it is higher than the 30%, then it means you are at the right end. But as you know, there is a 1 in 1000 error. So that means you need to get as high score as possible, just to stay on the accurate analysis, rather than having to find out that there was some sort of base call accuracy error. So now we will try to ascertain what actually each of these information that is available in our file actually means. So let's just make it a little short so we can compare it to the information we do have up at the top. And now you can compare this information. For example, at the top we do have the at operator, which basically starts with a sequence identifier, and that tells the program that is reading the FASTQ file that it is a FASTQ file. And then we have the instrument ID, the instrument that was utilized for the sequencing technology. And we have the run number, which can be numerical value only